This is lab number 12, biochemical testing number 3. This first video is just going to focus on the first part of lab 12. Before you start lab, disinfect your surfaces, wash your hands, and wear all the appropriate PPE.
So this is biochemical testing lab 3. So this is the lab in which we're going to use the large Petri plates. So you're going to need one with nutrient agar. So that's what I'm going to pour first. So again, you'll melt this in a way with 10-second intervals until it's liquefied. And then you're going to pour just enough so that it's going to cover the bottom of this plate. And this is the last nutrient agar plate that you're going to need to pour. So this is just making the yeast solution. So I have some warm water in there, about 230 mils. And then you're going to need to add some sugar. So we're just doing a tablespoon. So sugar is going to be the food source for this yeast. So a tablespoon of sugar and then a tablespoon of the yeast. You'll let that sit a little bit. You'll know if the yeast are viable and alive if you start to see foaming over this. So let this sit for 5 to 10 minutes or so, and you should be able to see some foam forming, which would indicate that the yeast is active. All right, so this is the inoculation of the nutrient agar plate for the last biochemical media experiment. So I have this one, and this is our larger plate, our about 90 millimeter plate, and I have them subdivided into 3 regions: a yeast region, a skin, and then in your procedure it asks you to do some local sample, and so I just chose the bottom of a shoe. I have my yeast sample that's been going for a little bit. You can see that it's starting to get a little bit foamy on top. There are bubbles forming, indicating the yeast is active, so that's good. I have some distilled water here because, like we usually do, we're going to moisten the sterile swabs prior to using them on the sample. So I'm going to do skin first. So just going to moisten that, and then forearm works just fine. So just rub that on, and as you're rubbing it, making sure you're kind of rolling it around back and forth. So rotating the swab as well as rubbing it. Okay. So then this is the skin region, and you're just going to do a zigzag. And as you're applying that, also doing a rotation of it or rolling of it. So next, I'm going to do the bottom of a shoe. So same procedure. Just going to moisten that a little bit. So this one shoe is going to be right here. All right, so the next procedure is a little bit different. We're going to get some of our yeast sample using a sterile pipette and a pipette pump. And you're going to do about 8 to 10 drops. And you're going to kind of rub that around. Then we're going to use a sterile swab. Trying to grab one of them to get a sample of it. Get rid of that glove and put a new one on. So this has our yeast sample on it. And this is going to incubate, and these are going to be the bacteria that you're going to use to prepare the test that's known as the Kirby-Bauer test. So that's going to be the next step, but this needs to incubate first. So like before, we'll parafilm this and let it grow.
Now that you have your plate ready, you can discard the yeast solution and you will parafilm your plate. Remember to take a photo of your plate, and then after that, it's just going to sit for 3 days, let it incubate. And then after 3 days, you will continue with part 2, which is a separate video.